Late in September 1982, an ordinary bottle of painkillers turned lethal, and 7 people in the Chicago area were dead within hours. A frantic FBI investigation, a trembling family kitchen table, and a quiet chemical clue would change how we think about safety. This is a story of pills and vials turned weapons, of forensic chemists, reluctant suspects, and the fragile supply chains that make modern medicine work. Before these headline moments, medicine felt like a quiet promise. You walked up to a pharmacy counter, handed over cash or an insurance card, and took home amber bottles and sterile vials stamped with lot numbers and expiration dates. Manufacturers like Johnson & Johnson and Baxter International and hospitals were the custodians of that trust. Regulators at the Food and Drug Administration and the Centers for Disease Control monitored safety, while FBI agents handled the rare criminal case. Then globalization quietly rearranged that trust. Starting in the 1990s and accelerating into the 2000s, the manufacture of active pharmaceutical ingredients moved offshore to factories in China and India. Intermediaries and brokers multiplied, and a single bottle of medicine could be the product of 5 different companies on 3 continents. Compounding pharmacies, like the New England Compounding Center in Framingham, Massachusetts, operated in a regulatory gray zone, filling shortages but also bypassing full manufacturing oversight. Hospitals came to depend on just-in-time deliveries, distributors like McKesson and AmerisourceBergen, and lot numbers to recall dangerous material. That complex apparatus worked most of the time, but it created new failure modes. Paper documents, forged certificates of analysis, and layers of subcontracting made fraud and sabotage easier to hide. In 1982, the Tylenol murders exposed that a bad actor could put cyanide into a consumer bottle and vanish. In 2001, letters laced with anthrax reached newsrooms, killing 5 and launching the Amerithrax investigation. Across decades, these episodes taught investigators 2 uncomfortable truths. First, some people will weaponize medicine for motives from money to malice. Second, the chemical and data trails those people leave are subtle but traceable if you know how to read spectra, shipping manifests, and laboratory notebooks. This story follows those traces and the people who read them. The first modern alarm sounded in hospital corridors rather than in courtrooms. Early in 2008, a nurse in a dialysis unit in Cincinnati reached for a vial of heparin, pushed the plunger, and watched a patient go pale and gasp. Alarms screamed, blood pressure plunged, and clinicians recorded the same description again and again in patient charts. Within days, similar clusters of allergic-like reactions and sudden hypotension were reported in emergency rooms in New Jersey and operating rooms in Texas. The pattern was uncanny because heparin is supposed to prevent clotting, not cause violent allergic collapse. State health departments and the CDC began logging cases and lining up lot numbers. By the time the FDA convened a lab team, the clinical picture was clear. Patients across many states had received the same drug lots and experienced a fast, complement-mediated shock, unlike ordinary heparin reactions. The forensic chemistry that followed looked cinematic in the lab. Under fluorescent lights, analysts ran nuclear magnetic resonance scans, high-performance liquid chromatography, and mass spectrometry on suspect vials. A senior FDA chemist later described how a hand-scrawled printout suddenly lit up with unexpected peaks. Those peaks were the signature of oversulfated chondroitin sulfate, or OSCS, a chemically altered cousin of a dietary supplement ingredient. OSCS was not heparin, but it could mimic some of heparin's physical tests, and it was cheaper to source. Crucially, the contaminated material triggered complement proteins in human blood in a way that caused the rapid hypotension and allergic signs clinicians were seeing. Within weeks, manufacturers like Baxter issued recalls on specific lots. The FDA posted advisories, and hospitals began quarantining vials. Investigators at the FDA, CDC, and academic centers worked like crime scene detectives, tracing invoices, following shipping manifests, and comparing spectral fingerprints until a clearer picture of deliberate adulteration and economic fraud began to emerge. Once the chemical signature existed, the story spread from spectra to shipping rooms. NMR printouts were a map, but investigators needed the roots. Epidemiologists matched adverse event reports to distributors and wholesalers, who pointed to manufacturers and then to raw material brokers. The trail moved offshore through chains of brokers and processors, and there, the motive and method became painfully ordinary. Public records and court papers show a pattern. Raw material sellers substituted or mixed cheaper chondroitin with crude heparin extracted from animal tissue. Then the chondroitin was oversulfated to increase its negative charge and to mimic heparin in some physicochemical assays. The result fooled many routine potency tests, yet produced a dangerous biological reaction in people. Regulators posted recall notices and press releases in March 2008 as more data arrived, and lawyers and prosecutors opened parallel inquiries. Investigators used enzymatic digestion with heparinases and then ran disaccharide mapping to show the wrong pattern of sulfation. They ran functional assays showing that contaminated lots activated complement in human serum. Behind those methods were people, a federal chemist who stayed overnight at an FDA bench and watched a waveform resolve into an unmistakable pattern. An epidemiologist in Ohio who linked emergency calls to a single lot number, and a compliance officer who found inconsistent certificates of analysis in a Midwestern warehouse. The human motive was often greed and gaps in oversight rather than a lone madman. Brokers chasing lower costs, weak inspection regimes, and opaque paperwork made it easy for a cheaper adulterant to slip into a life-saving drug. Meanwhile, the headlines were not only about heparin. Across the same decade, compounding pharmacies, ostensibly serving small patient populations, were operating with inconsistent sterility controls. That vulnerability exploded visibly in 2012. In late 2012, clinicians began to see a different horror. Patients who had received a steroid injection for back pain developed a strange meningitis caused by Exserohilum rostratum, a black mold. The cases clustered around a product shipped from the New England Compounding Center in Framingham, Massachusetts. By the time the CDC and the FDA linked infections to specific vials, the outbreak had spread across states, causing scores of infections and dozens of deaths. The scenes were wrenching. Family rooms in outstate hospitals filled with worried relatives, fungal cultures growing slowly in plastic trays, and state lab technologists watching plates fill with mold. Regulators moved to recall steroid lots, and investigators subpoenaed invoice books and environmental swabs from the compounding center. In parallel, the Amerithrax investigation that began after the 2001 anthrax letters still rippled through government labs. The FBI, under Director Robert Mueller, spent years analyzing spores, pore sizes, and genetic markers, and in 2008, the Bureau concluded that Bruce Ivins, a scientist at the U.S. Army Medical Research Institute of Infectious Diseases, was the principal suspect. That conclusion remains contested, but it underscores how insiders with access to biological agents or sterile products can weaponize them. The Tylenol case of 1982 kept returning in conversations among executives and public health officials. James E. Burke, the Johnson & Johnson CEO who ordered a nationwide recall after cyanide-laced capsules killed 7 in Chicago, set a standard many still invoke. But whereas the Tylenol attacker had inserted cyanide into bottles on store shelves, the heparin and compounding episodes showed a quieter, paper-heavy sabotage. Forged test certificates, false supplier audits, and layers of subcontractors meant an adulterant could be chemically engineered and dispersed without any break in a package seal. Investigators found themselves moving between frozen NMR printouts, a warehouse manager's handwriting, a compounding room swabbed for mold, and the inbox of a disgruntled employee whose emails hinted at motive. The escalation was not a single crescendo, but a chorus of failures. Criminal acts, corporate shortcuts, regulatory blind spots, and human malice all played a part. The pivotal moments in these stories came when a chemical signature met a paper trail and a human witness. For heparin, the clincher was an NMR spectrum that did not fit any authentic heparin fingerprint and an enzymatic digest that yielded the wrong disaccharide pattern. In a low-lit lab, an analyst slid printouts across a bench to a supervisor and said that the sulfate pattern proved this was oversulfated chondroitin sulfate. Within 48 hours, the FDA had notified hospitals, manufacturers issued recalls, and distributors pulled suspect lots from shelves. For the New England Compounding Center, the turn came when environmental swabs from a production room grew the same black mold found in infected patients, and shipping logs showed contaminated lots had been sent to multiple states. Legal responses followed quickly: recalls, criminal indictments, civil suits, and congressional hearings. In the Amerithrax saga, the FBI announced that genetic and forensic analyses linked the mailed anthrax to a flask in a U.S. government laboratory, and the Bureau named Bruce Ivins as the principal suspect. His death by suicide in 2008 ended a prosecution that many still debate. In the Tylenol case, the investigation never produced a murder conviction, but James Burke's decision to recall product and push tamper-evident packaging changed industry practice and federal law. Those decisive moments did not heal the families who lost loved ones, but they forced industry and regulators to change how they test, track, and respond. After Tylenol, federal anti-tampering laws and tamper-evident packaging became the norm. After the heparin crisis, the United States Pharmacopeia and the FDA added orthogonal tests including NMR to heparin monographs, and companies ramped up screening of active pharmaceutical ingredients. The 2012 fungal meningitis outbreak led to scrutiny of compounding pharmacies and to the 2013 Drug Quality and Security Act, which tightened oversight and created clearer pathways for recalls. The Amerithrax saga drove investment in forensic genomics and tighter laboratory access controls. Yet vulnerabilities remain. Global sourcing still depends on trust among suppliers, intermediaries, and regulators. Just-in-time inventories, opaque subcontracting, counterfeit markets, and non-physical attacks like the NotPetya cyber attack in 2017 that disrupted Merck show how fragile supply chains are to sabotage. The lessons are not only technical, they are organizational. Effective prevention requires better forensic capacity in public labs, mandatory serialization and provenance tracking, aggressive inspection of suppliers, whistleblower protections, and an industry culture where a single altered certificate of analysis sets off alarms. The orange pill bottle on a kitchen table and the soft plastic vial in an operating room are small objects that carry enormous trust. When that trust breaks, the consequences are immediate and human. The investigators who traced spectra to shipping manifests and spores to laboratory flasks closed chapters of pain. But their work also left an open warning. Modern medicine is only as safe as the weakest link in a chain that stretches around the world.